Organisms live in different places because the environment varies in factors such as temperature, moisture, light, soil composition, and the availability of food and shelter. These variations create a range of habitats within an ecosystem, each suitable for different species. For instance, some plants thrive in sunny, dry conditions, while others prefer shaded, moist areas. Similarly, animals may be found in areas where their specific needs, such as food sources and nesting sites, are met. The distribution of organisms is therefore not random but is influenced by the specific environmental conditions of each location. To study the distribution of small organisms, such as plants, insects, or other ground-dwelling species, ecologists often use a tool called a quadrat. A quadrat is a square frame, usually made of wood, metal, or plastic, with a known area, such as 1 square meter. The quadrat is placed randomly or systematically on the ground within the study area, and the organisms within the frame are counted or recorded. By repeating this process across multiple locations within the study area, scientists can gather data on how organisms are distributed. This method is particularly useful for studying stationary or slow-moving organisms, as it allows for a snapshot of the population in a specific, defined area. The use of quadrats provides a standardized way to collect data, making it easier to compare different parts of an ecosystem or to monitor changes over time. For example, if researchers want to compare the distribution of a certain plant species in sunny versus shaded areas, they could place quadrats in both types of locations and record the number of plants found in each. This data could then reveal patterns in how the plant species is distributed relative to light availability, helping scientists understand the environmental preferences of the species. In addition to studying distribution, quadrats can also be used to estimate the population size of an organism in a particular area. This is done by placing multiple quadrats randomly within the study area and counting the number of individuals of the target species within each quadrat. The average number of individuals per quadrat is then calculated and multiplied by the total area of the habitat to estimate the total population size. For example, if a researcher is studying a 100 square meter field and finds an average of 5 plants per square meter using quadrats, they can estimate that the field contains approximately 500 plants. This method provides a practical way to estimate population size without the need to count every individual in a large area, which would be time-consuming and often impractical. However, the accuracy of the population estimate depends on the number of quadrats used and their placement within the study area. The more quadrats used, the more reliable the estimate will be. Additionally, random placement of quadrats helps ensure that the sample is representative of the entire area, avoiding bias that might occur if quadrats are placed only in areas where the organism is particularly abundant or rare. In summary, organisms live in different places because environmental conditions vary, creating distinct habitats that support different species. Quadrats are a valuable tool for studying the distribution of small organisms within these habitats, allowing ecologists to collect data on where and how many organisms are present. By using quadrats, researchers can also estimate the population size of an organism in a larger area, providing insights into the abundance and distribution of species within an ecosystem. This information is crucial for understanding ecological patterns and making informed decisions about conservation and habitat management.